IB Bio, Option B, Biotechnology, Bioinformatics Part 8 is the second of two movies that look at B.4, Biotechnology in Medicine. Part 8, B.4b, this movie, will have its focus on the use of biotechnology in the diagnosis of disease and treatment of disease. The essential idea is biotechnology can be used in the diagnosis and treatment of disease. Here is the outline of all the movies available for Option B. Use this outline to find the movie you need for review. This movie is focused here, but this movie covers more than just treatment of disease. Part 8 examines diagnostic techniques related directly to DNA as well as the use of biotechnology in providing therapy. So let's jump right in with the first IB syllabus statement. Describe the use of PCR to detect viral infections, specifically influenza. In the way that ELISA detected small quantities of antigen or antibody in the blood, as I covered in Part 7, PCR can be used to detect the presence of pathogenic DNA or RNA. Let me say this again. ELISA diagnoses disease by the presence of the pathogen's antigens or antibodies against the antigen in the bloodstream. PCR detects the presence of DNA or RNA of the pathogen. You might say pathogen RNA? Yes, many viral pathogens are RNA viruses, retroviruses. Influenza and HIV are retroviruses, RNA viruses. The second statement, polymerase chain reaction, serves to amplify minute quantities of DNA. The use of PCR to detect viral infection provides an early diagnosis of viral infection. For people with compromised immune systems, determining the presence of the virus at an early stage of infection is critical to their treatment. A DNA virus will infect a host cell, as can be seen in this image. The DNA becomes incorporated into the host DNA. Testing host DNA for the presence of viral DNA would detect viral infection, and this can be done. Cells of the host can be sampled, and the viral DNA can be detected using PCR. Let me repeat, cells of the host can be sampled, and the viral DNA can be detected using PCR. Pay close attention in moving forward. An RNA virus, a retrovirus, will infect a host cell. The viral RNA is copied into DNA using the enzyme reverse transcriptase. The copied viral DNA is incorporated into the host cell genome. Testing host cells for the presence of viral DNA or RNA would detect viral infection. Cells of the host can be sampled, and the viral RNA or DNA can be detected using PCR. Stay tuned. Examples of RNA viruses, also known as retroviruses, are the influenza virus as well as HIV. The glycoproteins seen here on the surface of the influenza virus are the antigens that might be detected in the blood using ELISA. But remember, viruses get inside cells. They infect cells, making detection difficult. Host cells, infected cells, can be sampled for the presence of viral RNA or DNA using PCR. Remember that retrovirus RNA is copied into DNA by the enzyme reverse transcriptase. This reverse transcription happens in the cytoplasm of the infected cell, the host cell. Now, these host cells, the infected cells, can be sampled for the presence of viral RNA or DNA using PCR. Cells from the infected patient are sampled. All the RNA from the cells is extracted. A primer sequence specific to the influenza virus is added, and the primer is the key step. The primer targets base sequences specific to the virus. The primer is a probe, like a needle in a haystack. The primer binds to viral RNA only, not host cell RNA. A DNA copy is made using reverse transcriptase, but only viral RNA is copied into DNA because of the primer, and PCR amplifies the viral sequence that can be detected by gel electrophoresis. Here is a scanning electron micrograph of the influenza virus. Influenza virus is an RNA virus, so the primer targets viral RNA before reverse transcriptase copies the viral RNA into DNA. Once PCR amplifies the quantities of viral DNA, detection is possible. One more time, host cells are sampled, RNA is extracted, including RNAs of the host. The primer targets specific viral RNA, then reverse transcription copies only the viral RNA into viral DNA. Then, only then, is PCR employed to amplify the viral DNA for clear detection using gel electrophoresis. Here is the method once again. I'll let you study the details of this slide on your own. Remember that the primer is constructed to target whatever strain of influenza might be of interest, bird flu, swine flu, or other. That said, the IB syllabus focuses on PCR as the principal technique. The syllabus statement is describe the use of PCR to detect different strains of influenza virus. Here is another use of the PCR. Take some notes here. The PCR could be used to detect or rule out a rare disease or illegally caught sold species. So for example, take samples from a species about which more information is needed, the unknown. This could be a disease or sample of a fish from the market. Construct test primers, primers from known or legal species. The primers could be from genes for cytochrome C or 18S ribosome. Run PCR, and if no amplification occurs, the unknown species is not known or not legal. You should remember the basics of PCR from your earlier study of biotechnology. You need a primer that targets the sequence of interest, the viral DNA formed following reverse transcription. For PCR, you need the nucleotides, the Taq polymerase, and the DNA sample, which in this case would be the DNA copy of the RNA viral sequence. Here are the steps in amplifying the DNA. You should be familiar with these steps from your earlier study of biotechnology. I'll let you study this slide on your own as we bring the use of PCR to detect different strains of influenza virus to an end, and we move on to the use of another technique to detect DNA sequences of pathogens.